Malaria, a terrible disease. It's caused by a parasite that is carried by the female mosquito, and it kills over 1 million people every year, and that's despite the availability of drugs that can treat the disease effectively. A major problem now is that the parasite has developed resistance to the classic chloroquine drugs, which have been very effective over the years. Now, this was already a problem during the Vietnam War when North Vietnam asked China for help. China, as you know, has a long tradition of using plants to treat disease, mostly based on anecdotal evidence. In any case, pharmaceutical chemist Tu Youyou took on the task of combing through ancient texts to look for any mention of treatments of malaria. And she found repeated mentions of Artemisia annua, commonly called sweet wormwood. There's a plant from which her team isolated Arteminisin, a compound that indeed proved to be an effective treatment for the disease. Arteminisin saved millions of lives and garnered Youyou a share of the 2015 Nobel Prize in Medicine. While Arteminisin is an effective medication, its extraction from a plant is a complex process and yields are low. This makes it an expensive medication, unaffordable by most people in countries where malaria is a problem. Science, specifically synthetic biology, has stepped in to help. Synthetic biology refers to the manipulation of living organisms to produce a desired outcome. In this case, that desired outcome is the production of Arteminisin, and the organism that is manipulated is a bacterium or a yeast. The first question researchers asked is, how does the Artemisia plant produce Arteminisin? Plants, you know, are veritable chemical factories. They take water, carbon dioxide from the air, and minerals from the soil to produce all sorts of compounds upon directions from DNA, the proverbial blueprint of life. Genes in DNA code for the production of enzymes, which are specific proteins that act as catalysts for the chemical reactions that piece together molecules. Amazingly, the genes that code for the enzymes needed to make artemininic acid, the plant's precursor to Arteminisin, have been identified, and they've been inserted into the genome of a bacterium or a yeast. When these organisms are fed glucose, they produce artemininic acid that then, with a simple chemical reaction, is converted into Arteminisin. What human ingenuity can do is just amazing. Those microbes that have been cleverly manipulated by synthetic biology are now saving millions of lives. And that for today is our Kapajao.